So welcome back. I trust you got a little refreshment during the break. We are going to buckle up now because we're going to change into the rapid fire presentations that we have planned for you. Or I don't want to say we because somebody else has been forced to plan well ahead in order to make a quite swift presentation of their research and what they're looking at right now. These are all, it's 4 EU cancer mission projects that will be presented with. And each project gets around, well, I've set 3, but if they stretch it, I'll allow 4 minutes and they represent various aspects of the mission, ranging from coordinating funding to molecular profiling, immunotherapy, and early detection. So we're going to do them one by one, and it's going to go fast. So as I said, buckle up. The first one we'll hear from, I hope they are ready to take the stage, is, here we are. Here's Nancy. Nancy Abu Zaid is starting her screen sharing, and I'm sure she will be ready with us right now. She unfortunately couldn't be here with us today, but she, as you can see, is going to join us online. So do we have Nancy ready with us? I can tell about Nancy that she represents the FORCE project, fostering oncology research by charities in Europe. And Nancy Abu Zaid from the Fondation Arc leads this mission project coordinating charitable cancer research funding across Europe. And I think we have Nancy with us now. Do you hear us, Nancy?
Yes, do you hear me?
Yes, we certainly do. And I'm going to start your 3 minutes now. So okay. Go ahead.
Thank you. Thank you. So I will talk about FORCE, which is a unique initiative by charities to foster pragmatic clinical research in oncology in Europe. Next slide, please. So you have here, I'm representing a consortium, 14 charities from 11 European countries. You have the name and the map here. And we hope that during the 4 years that we will be funded by the EU, we will manage to grow even broader. Next slide, please. So FORCE is a program that will allow us to define and deliver a pragmatic clinical trial program by cancer charities and dedicated to rare and hard-to-treat cancers. During the process, we will implement 2 transnational call for proposals that will allow us to select and fund clinical trials over Europe. And at the end of the project, we will deliver a blueprint on this topic to the EU, hoping to contribute to future decision-making on this topic. I will not go into the detail of the way we are working, the work packages you have on the right of the slides, but just to let you know that we have a dedicated work package to patient centricity and impact, because this is a central issue for us. We want to involve patients at all stages of the project, from the definition of the program to the call for proposals that we will launch, and to the project, to the clinical trials that we will fund. Next slide, please. So just a few words to let you know what are pragmatic clinical trials. These are typical clinical study designs that help evaluate effectiveness of an intervention in a real-world clinical practice. So usually they have a very broad eligibility criteria that fit real world. They compare interventions to standard practice. They apply a very flexible delivery of treatment, as the treatment should be available anywhere. They have very relevant outcomes that focus on patient needs and patient-centered outcomes, and they integrate with routine data, with registries, with electronic health records, etc., etc. Next slide, please. So why do we as charities are interested in this type of clinical trials? Well, as I said, these trials have patient-driven priorities, and this is central for us charities. They provide faster and affordable evidence. They tackle the issue of access and equity, as the treatments or the interventions should be available in smaller centers and underserved regions, and not only in the very big specialized cancer centers. It's usually independent from industry, and it will help us on strengthening our advocacy, as the results that will be made available will help us to influence guidelines and shape policy. Next slide, please, and it's my last one. So if this topic interests you, don't hesitate to contact us or even to join us. We are looking for partners, charity funding in Europe, or partners in cancer centers, clinical units, patient organizations, or other types of advisory partners like policymakers, regulators, etc. So don't hesitate to visit our website. We are active on LinkedIn, or you can send me an email if you have any question. Thank you.
Nancy Epizaid, thank you. So next up is Professor Harold Mishak of Multir, Molecular Profiling to Guide Bladder Cancer Treatment. And Herr Mishak, guten Tag.
Thank you very much. Nice to see you.
Please.
Okay, so I represent Multir, a project on tumor host interaction. And why do this matter? A tumor needs to grow and thereby interact with the host the whole time. Tumor obtains nutrition from the host, but also has to protect itself from the host, from immune response, for example. Immunotherapy was a revolution, but it only works for a subset of patients. We do not know how immunotherapy exactly works, what triggers the response. And therefore, we generated the hypothesis that there must be generic, let's say, proteins, biological variables that determine the tumor host response and determine the interaction of the tumor with the host for any type of tumor. On the next slide, please. We are a consortium of 11 European countries plus Canada, and we decided to go for melanoma, lung cancer, and bladder cancer because these are tumors that are to some degree responsive to immune therapy, to checkpoint inhibitors, and we examine host factors that may be age, sex, genetics, but also different omics data, especially proteomics, transcriptomics data. And in doing so, we could in fact define first relevant interactors of the host with the tumor, some tumor-agnostic potential predictors. On the next slide, we see some of these, or let's say the nature of these, and this is to a large degree the extracellular matrix. The extracellular matrix is in fact a conglomerate of different proteins that form this matrix, and the tumor cells need to interact with this matrix in order to grow. The matrix guides immune response, but it also protects the tumor from immune response. We have by now also technologies available to assess the extracellular matrix quite efficiently via the analysis of peptides and proteins in body fluids, and we can link this back to the tumor, to the immune response. And in the last slide, we then found that several of the, can I have the next slide, please? Thanks. We found that several of these proteins and peptides from the extracellular matrix are associated with outcome and appear to be also associated with response, can predict outcome and likely can predict response. Many of these collagen, CD99, others also surface molecules that interact with the extracellular matrix. And this was also confirmed using other omics technologies. So ultimately, what we see already developing after 2 years, which is quite a good success, is that we can answer some of the questions that by understanding tumor-host interaction with the extracellular matrix, we have a very good target to interfere. So this was a success story. And last but not least, I also want to say that we spent the majority of our efforts, in fact, on legal, bureaucratic issues. And this is, if these would not be present, I think we could be at least twice as efficient and twice as fast. Thanks.
Thank you. So who would have thought that this quick round would also contain a bit of political activism? That's quite refreshing. Thank you for that. So next up is from Pragmatil, Dr. Vladimir Galbao de Aguiar. And Pragmatil is on advanced immunotherapy using tumor-infiltrating lymphocytes. Please.
Thank you for having me. We know that the TIL therapy has a lot of activity in melanoma and other types of tumors, but we know that we need to expand the indications and adopt in different other centers, not in research centers. So we know that one of the aspects that limited the expansion of this therapy is the toxicity. So the Pragmatil trial addressed this, replacing the high dose of interleukin-2 used adjuvant after the TIL infusion to a new interleukin-2 that is less toxic, that was tested in phase 1 clinical trial. And we use that with this consortium. It is composed of 12 partners, the consortium, in 6 countries. We lead the project, and the design of the project is to include 40 patients in 2 arms, 20 patients in each one. The first arm, we will include patients with the classical TIL therapy with high-dose interleukin-2, and the other arm, we will replace this interleukin-2 to the Anaveon ANV419. Next slide. So the primary objective is focused on safety and quality of life. So we will evaluate whether the TIL ACT using this IL-2 analog compared to the previous slide, please. Yes. Is capable to reduce the mean number of predefined grade 3 or higher relevant adverse events related to the interleukins and also improve patient-reported outcomes. Next slide, please. Next slide. We have 6 work packages, and I will highlight here the work package 3 because it is centered in the patients as co-researchers and evaluates the short and long-term patient-reported outcomes. The objectives are to ensure patient involvement through the whole project life cycle and to capture short and long-term treatment-related toxicities and quality of life. Next slide, please. So to get there and give voice to other Andreas in this clinical trial, so we developed a methodology of action plan, system-oriented dialogue model. We did living labs with the patients to train them and perform their roles also as co-researchers from the beginning of the clinical trial. Next slide. Thank you very much.
Thank you. Professor Michael Akian from Endeavor Early Detection, and I have struggled to pronounce this word all my life. Esophageal. Esophageal. That's quite a profession you have here.
Difficult words. Adenocarcinoma.
Oh yeah, that one. That one exactly. Please.
Thank you, Mette, and thank you to the EU Cancer Mission. I'm gonna try to sum up what we do in 3 minutes at the Endeavor Consortium. It's 15 partners, academic partners, 2 general hospitals, and 2 industrial partners. And like Mette taught us all, you have to start with the conclusion in the beginning. So we're working. Our goal is to optimize the surveillance strategy and to detect early recurrence of esophageal cancer. Next slide, please. So, what is the current problem? Our current problem is that a precursor to esophageal cancer, Barrett's esophagus, and early esophageal cancer we can treat endoscopically, but we have no idea who are going to have a recurrence of the cancer and who is going to stay in remission. This leads to a high frequency of follow-up for all patients, which again leads to decreased quality of life for patients because they have to be controlled many times, increased healthcare costs, and a lower patient-centered treatment algorithm. So our purpose was to try to implement a risk stratification method to individualize follow-up for Barrett's esophagus and for early esophageal adenocarcinoma. And we aim to do that by doing brush cytology, biopsies, and blood samples. And by analyzing with DNA fish and next-generation sequencing, we hope to achieve this goal. We also send out questionnaires for patient-reported fear and anxiety related to treatment, and we heard that from Andrea earlier this morning. That is actually quite cumbersome for cancer patients to undergo these diagnostics and follow-ups. So currently we are in a pilot study, and we have included 8 patients out of 60, and we're trying to work out the logistics, like we heard, with all the permissions and bureaucracy. And once we have finished the 60 patients pilot, we're going to go on and start the RCT, which is 200 patients. Next slide, please. So a very busy slide here. It's the timeline of our trial, and we have it's 3 years for the RCT, and as you can see on the slide, those who has complete eradication of their Barrett's esophagus, we only do endoscopy once every year. But those who has remaining Barrett's, we do every third month, and try to eradicate the Barrett's. In the intervention arm, we have a lot of controls, but that's to ensure that we catch those who has a high risk of recurrence. And the end, like I said, is trying to make a strategy where we can surveil those who recur at a higher frequency and those who doesn't at a lower frequency. That's it.
Thank you. I think we should congratulate all these people on keeping time and teaching me how to say esophageal adenocarcinoma. Thank you for that. So we're ready for the panel number 2, which is on cancer immunology and biomarkers of immunotherapy. So we've already heard immunotherapy mentioned several times today, and it has transformed cancer treatment. This is, some would say, maybe the greatest success, one of the greatest success stories of modern oncology. Yet it seems to be working, as it seems to be working brilliantly for some people. We've also heard it doesn't work for some other patients. And the cancer mission recognizes that the next frontier is, of course, precision immunotherapy, predicting who will benefit and how will they benefit, and understanding why some tumors seem to resist and develop strategies to overcome that resistance. So that's what we're going to talk about now. So we're going to start the panel just like we did before with co-creating a word cloud. And we'll ask you the following question. In one word, and you did that really well last time, so let's just keep up that good work. In one word, what do you think is the biggest challenge in translating immunotherapy research into personalized treatment for individual patients? What is the biggest challenge in translating immunotherapy research into personalized treatment for individual patients? Or in other words, take the next step, as I just mentioned, the next frontier. Money, yeah. So it takes money, but is money actually the biggest challenge? Yeah. Oh, scaling, precision, knowledge sharing, biomarker, lack of pragmatic trials. That was 4 words, but interesting anyway. Advocacy, collaboration, knowledge sharing and collaboration, I see go together. ethical committees, patient engagement, regulations, sensitivity, funding and money. I mean, if you put those two together, they take up the whole screen, don't they? And patient engagement is quite large as well. I'm a bit surprised about that. Political willingness, yes. Lack of pragmatic trials. I think you're pivoting into giving more than one word. Just, I think it's because I praised you, and now you think, okay, I can do 2 words or 4. It does work better if we, because we're not gonna get those big words if people write more than one word in one entering. So, okay, biomarkers, scaling, right? Okay, well, I would like to invite my panel up here now, and we have with us Professor Marek Svoboda. We have Dr. Robert Alexandrovich, Professor Nikolai Juhl-Bierbach, and Vladimir Galbao de Aguiar that we just heard from. Please join me up here, and you may welcome them on stage. And do we have Professor Svoboda with us online? Nope. Mr. Svoboda is not currently with us, and we'll see if anything changes in that regard. Okay, but did you write a word? Yes. What word did you write?
Money.
Money. Money, money, money.
That's the beginning and the end of everything.
Yeah. Do you like money for your research?
Like everybody, there is no translation of the research without the money.
Right.
I would like to explain this later on.
I'll let you. What did you write?
I wrote scaling.
Scaling.
So yes, if you want to really leverage like the new advances in AI technologies, we need large amounts of patients, large trials, large cohorts.
Right. What does that require?
That requires the courage to go beyond the small cohorts that we usually work in with a couple of hundred patients to be able to assemble hundreds of thousands of patients. Mm-hmm. Mm-hmm. And that require multi-center approaches and cross-border collaboration to a much higher degree than we are doing today. And as has been mentioned several times now, that requires some laxing on regulatory approaches, or at least to provide, to facilitate ways where we can more easily share data and samples between countries and collaborators.
Collaboration.
Collaboration.
Yes.
Yeah.
For me, it's very important.
With other projects and other researchers around
not
Europe.
other projects, but in the same center sometimes, because usually we have people from the preclinical, clinicals, we have regulator bodies, and sometimes people have an excellent idea, but nobody knows that. So collaborate and keep, make a room for discussions. I think that's a very important thing. So,
Mr. de Aguiar, I think I'll start with you. We just heard you present swiftly what your project really. Mm-hmm. So, if we were to start with the challenges that you're touching on a little bit here, do you want to start out with just for an initial presentation of how do you see those challenges and how is it affecting what you're working on right now?
Well, the challenge in the Pragma-2 trial is to put together different countries in the same project, because we have some institutions that have more experience, others with less experience into therapy. So some of them are more advanced in some issues, others are less, but we needed to put all of these people together to make a project.
Right. Nikolai Bierbach, if you were to, for an opening statement, I mean, you're talking, we did touch a little bit on it already, but just for an opening statement, just telling us where are you right now and what is the project you're working on currently?
So I had a translational cancer research lab in Aarhus University, where we do bioinformatics research, so it's all computational. That's like AI approaches, so that's why we like to scale
indicated.
to large trials. And I worked a lot in the biomarker space. I published, I don't know, 20, 25 papers on different biomarkers, and only one of all of those actually made it to the end. And that took more than a decade. Now we are focusing much more actually on more holistic approaches. So we used to focus on the tumor itself, as many others try to develop tumor-specific biomarkers. But recently, moving towards immunotherapy, we have been approaching what is the state of the patient's immune system? How does this actually impact immunotherapy response? In that regard, we published just early this year, actually, a study where we've investigated the circulating T-cell landscape and found that there is a considerable variation in what is the immune health of the patient, and this actually impact the response. A patient with a poor state, some few T-cells, low diversity perpetuas, have, in this relatively again smaller study, more than 5 times higher risk of death from this bladder cancer.
And of failing that treatment, right?
Yes, indeed. So this wasn't immunotherapy, this was a standard of care. But taking that further, we were now looking at more the fundamental approaches. So where does the T-cell landscape come from? They come from this organ called the thymus that is understudied, really. And this has shown that the immune system decays with age in everyone, but it decays on different rates. So not everyone decays at the same time. The thymus stays active into your 60s, maybe even longer. And those that respond better to immunotherapy seems to be those where the immune system works better in the first place. And I found these angles are really interesting. I think combining that with tumor-specific biomarkers as currently in use, PD-L1, TMB, that has significant room for improving patient treatment and outcomes.
Very interesting. Thank you. And Mr. Alexandrovich, just for an opening statement, and you can touch on that money thing as well if you want, but let's just hear what it is that you're working on currently.
Okay, so I'm doctor of bioeducation, but actually professionally for 20 years I'm building the companies. I'm serial entrepreneur. I built more than 10 companies
Good
in
for you.
medtech and biotech area. So I'm like a Bob the Builder, maybe, this kind of the things.
There isn't much resemblance, I must say, Dr. Alexandrovich.
Yeah. So I'm talking about the money because of my profession, and I see how the right funding is necessary for the translation of the great research into the reality. Because we can talk here about the great projects and the great scientific publications or approaches, but finally, all what we are using in the hospital is brought by the companies. So at the end, each research to work for the patients has to end up in the technology which is produced and supplied by the companies. And this translation is absolutely necessary for any kind of the research to be viable. I will give you an example. Europe has an excellent research still, but the others are catching up. China is catching up very quickly. The Middle East is catching up. Still, 4 of the 5 best-selling products were scientifically discovered in Europe. Keytruda, Ozempic, Comirnaty, all these names you know, everything was invented here. Even it's somehow marketed and sold by the others. One element which is important in this breakthrough therapies is really discovery, and Europe is very strong in this. And for example, in our company, which I'm right now heading, we are also starting the technology transfer from the deep research. The company was founded on the groundbreaking discovery of Professor Krull. A few years ago, she discovered new biological mechanism which was unknown. This is a mechanism which is called TRAIN. It's a transfer of iron-binding protein. Half of the tumors is consisting of the macrophages, and macrophages are transferring ferritin very quickly to the cancer cells. And we use this to create completely new class of the therapeutics. How it was perceived by the European Union, I would say that Professor Kuhl has received 3 times ERC grants. It's quite exceptional result. Only few people in Europe received 3 or more grants behind the discovery. And it was also included as one of the top 10 most important discoveries in the 10th anniversary of the ERC. So this is our beginning, the groundbreaking research and the groundbreaking discovery.
So say a little bit more about that. I mean, why is it that this maybe can work better than the current immunotherapies?
Well, definitely. I mean, we are talking here about the personalized approach, but we have to define what does it really mean, the personalized approach. A few years ago, I have heard the speech of the chief of the Catapult, the UK accelerator, and he said that he had a vision that in every hospital there will be a machine which will create the personalized therapy for the patient. Like it was the beginning of the CAR-T. After a few years, he said, no, it will not work. It's too costly. And this is why I'm talking about the money, because we have to keep in mind this, that at some point, the cost of the therapy could be a barrier for introducing its global. It was the case with the CAR-Ts, which are excellent things. What we are doing, we are actually having the personalized therapy, but in a completely different approach. With the train mechanism, we have created macrophage drug conjugates. What we made, we replaced the iron in the ferritin. This is the protein which is transferred from the macrophages. And we created the Trojan horse. Everybody knows what the Trojan horse is and how it worked.
Yeah. Somebody was inside.
Exactly.
To fight the ones
with this
that
train, we are saying that we have changed the passenger and nothing else. We are not changing the cells. We are not manipulating the macrophages. We are just giving the macrophages inside the tumor. And this is why I'm saying about the intratumoral approach.
So you sent stronger soldiers in there, so to speak.
That's right. That's correct. We are using 1% of the drug dosage that is normally used in ADC formulations, which is very small. So we are avoiding a lot of systemic toxicities. Our drug is actually not detected in other tissues. But what's happening is the pure biology. The macrophages are transferring the ferritin with the drug. The cancer cells are killed. And the macrophages are the only cells which are penetrating very deeply to the hypoxic area. They are cleaning the debris. So they are starting to present these antigens of the killed cells to the immune system. And they are activating tumor-associated macrophages. We were talking here about tumor microenvironment. Everybody is complaining that something works, but the tumor microenvironment is protecting it, is not having the enough efficiency. Here, the macrophages are changing tumor microenvironment because the tumor microenvironment are the macrophages, are the colleagues. So they are feeling like at home. You can imagine they may be seeing the Sweet Home Alabama. They love it. And they are reshaping, reprogramming the immune system. What we have seen, it's absolutely activation of the immune system. Where we are now, we do not see it just in the Petri dishes, in the vitro experiments. We have made a number of preclinical experiments in different solid tumors. The most difficult, we were talking about the pancreatic cancer, yes, platinum-resistant ovarian cancer, or brain cancer. In brain tumor, in glioblastoma, in animals, we just gave 2 shots and we obtained complete eradication, complete tumor removal and cancer resistance in most of the animals, in more than 90%. 2 shots, nothing else.
That sounds groundbreaking.
That's groundbreaking, exactly. But what's the origin? The origin is, again, I say, it's the discovery. It's the same story comes again. Like Fleming, who has discovered the penicillin. It's like DNA discovery. Everything starts from something which is in nature, and we are coming out from the nature. How we can translate this into meaningful therapy or meaningful procedure.
And a long road ahead before that will be injected into a human being, right?
No, it is no. We are actually, we have, this was discussion about collaboration. Actually, this is, we have established the manufacturing process across the Europe. We are collaborating across 8 countries in Europe in establishing the process, the quality. We are ready to start the clinical trials. But again, the money is the problem. And this is the European problem. I will give you the example of this discrepancies in the money. In 2024, so 1 year ago, US had the funding of approximately 210 billion euro for all the biotech companies. At the same time, in Europe, it was 57, so 25%. This is the gap, which is absolutely not fulfilling the needs of the science that we are creating in Europe. So there's excellent funding of the research, like from ERC, EIC programs, from here what we are seeing, but then it's a gap. And that's the biggest problem of Europe right now.
Well, fortunately, we're sitting here right in the center of the cancer mission, so so far so good, right?
Well, so far, it's good that we should shake. I think the first really flag and the notice was 1 year ago when Mario Draghi made the report, and it was a bit shock. And I'll give you another number. Last year, transactions, because all the small startups who are creating the products, they will not market these products finally. Usually they will sell the products to the big pharma. This is the normal process. 40% of the transactions last year was between big pharma and China. 5 years ago, China transaction level was 0. Last year, transactions with Europe was only 15%, and it's declining. Again, why? Because China is covering this process not only of the discovery. They are worse than Europe in new chemical entities, in new classes, but they are very quick in upgrading what is currently on the market. And they are covering this clinical setup. And this is the moment when the big pharma is coming and saying, okay, I will buy your product because you have proven that it's working in the humans. That's the problem.
So let it be known that we're not just interested in reducing bureaucracy, we're also interested in increasing the money tank aiming towards cancer research. So now we have the political statements on set, right?
Yes.
Good. Getting back to the immunotherapy, I want to call it a paradox, which is we have this revolutionary approach to curing cancer, but it works sometimes, and then sometimes it doesn't. And if we talk about your work, Professor Bierbach, on predicting the response of the immunotherapy, and you were talking about scaling because right now you're within this limited area, but what is the next step then? If I were, let's say I'm a patient, I want to know when does it come to my end? When does it target my kind of cancer? When does it target my type of patient?
Well, so
what's
I
the
work
answer?
in the discovery phase, which is
Yes.
you will get your drug when it has been through your process. But in the discovery phase, we are limited by the size of the trials often, so we can have excellent hypotheses, and we can test some of those.
Yes.
And we can find a biomarker often if you give us a well-powered cohort, which rarely happens. It's often we work in a couple of hundred patients that has received a drug. Some of these respond to that drug. The data-driven approach would be that we go and we try and compare these two sets of patients. What makes the responders stand out. And then we can make a model that encapsulates this. Where it often fails is that we need to validate this finding, of course.
Right.
We can build a model that will predict its response, but it's often, given the small size of the trial, the cohort, it's often overfitted, or it finds spurious results that only is true in this cohort. So validation is the next step, and this is often the step where like more than 90 and 95% of all of our biomarkers will fail at this step. But if we are lucky, and I think this is very much about, it's about luck, but it's also about, of course, if you have multiple different cohorts from different sources, you may be able to identify something that is more biologically real. And then we can take it to the next phase, which is validation. You can have an independent cohort, properly sized, where you can validate it. If it validates, you can publish. And this is where my work usually ends. And then the biggest obstacle to bringing it into a corporate setting, at least from my experience, is that many of these biomarkers are not immediately patentable. So it's not necessarily either the researchers didn't think about it, this has happened to me as well, or the approach just, it's not something that you can patent immediately. Like at the moment, for example, you cannot look at the thymus. Anyone can look at the thymus. You can do it as well, it's not patentable. It's real biomarker, but how will you bring this to market? Because there's no company that will pick this up, as there's no
And pour
IP.
money into further developing it without being able to actually
Indeed.
secure
So-
its investment.
From discovery to publication, that could be a 2 or 3 years process. From patent to product, again, this is your field, but it's at least 10 years. Lots of work and a lot of money. Usually, I think it's hundreds of millions of euros, right? You would want to pour into this phase. And that's often where it breaks down when it comes to the individual patient. So if you're asking a patient, when will this benefit me? That's very far into the future.
I think we finally succeeded in getting a connection through to Professor Marek Svoboda. Professor Svoboda, can you hear me?
Yes, I can hear you, and I'm so sorry to be late and for some technical issues which I'm now overcoming, and I can be with you.
We'll blame it on the technical problems. Don't tell anyone you were late.
Yeah, sometimes it happens.
We started out doing a word cloud here, and we have been discussing the problems of scaling. We've been discussing the problem of money, and I think we kind of closed that. But I've also had the other panelists just make a brief opening statement on what it is they're working on right now and what their main challenges are. So why don't you take just a couple of minutes to just let us in on what's going on with your research right now, what are the main challenges that you're facing right now?
Well, I'm a medical oncologist and clinical geneticist, so I'm dealing in my outpatient office with cancer patients on a daily basis, and of course, the immunotherapy and this area is very important for us because, and I'm pretty sure it was already mentioned, the immunotherapy really help us to treat patients and to change the situation and to help to make really new patient cancer survivors, also with the cases who are metastatic or very, very advanced. So in my case now, it's really the topics which I'm very interested as on the research level, from the personal perspective as an oncologist, and the research level now is the aspect of the how the further use of immunotherapy, how to overcome the immunotherapy resistance, in particular how to transform initially unresponsive cold tumors into hot tumors that responded to treatment, and of course the identification of biomarkers of severe immune-related adverse events with immune checkpoint inhibitors and its possible prevention, and especially with non-immunosuppressive treatment of such symptoms, because the side effects are really a very important aspect here. As I mentioned, this treatment has the potential to produce the new patient survivors, including the long-term one. So that's from point of what I'm doing my my oncology practice and the research which is done in our National Cancer Institute. And of course, I have some role in the Cancer Mission Board, which helping to give the advices to relevant persons in DG RTD and DG Sante about what is ongoing and what is needed.
And when we're talking about, I mean, if we're talking about that gap in ensuring that the discoveries actually turn into solutions for real patients, the barriers that you're addressing now, can you elaborate a little bit more on those barriers and maybe even touch on how the Cancer Mission helped to overcome them?
Oh yeah. Well, first of all, regarding the barriers, yeah, the Cancer Mission really certainly helps to overcome barriers, especially in the biomarker research. At the very least, it has a clear enemy, which is the cancer, and the Cancer Mission used the scientific framework as a strategy to fight this cancer. And importantly, it has a financial tool in the form of a number of Horizon Europe calls for proposals on cancer research to support the EU mission on cancer in all the pillars, which are not only the understanding of the cancer, but also the prevention, diagnostic, and the quality of life, as I mentioned. So in addition, the Cancer Mission also contributes significantly to the potential secondary use of high quality data, which is very important, the high quality data from real world clinical practice, which is a very important barrier in implementation of the innovation, and of course, including the harmonization of the collected data. What I would like to partially mention, for example, is the Juncken EU data platform project. Our institute participated also on this project, which is also one of the Cancer Mission flagships, which enable researchers to work with the data, different sources, and also in the huge scale, which is very important. And it will for sure improve the understanding of the cancer. Another thing is, another example could be, for example, the Cancer Mission supported project CCI for EU, which aims to find ways how to build a comprehensive cancer infrastructure around existing cancer centers in the member states, which is, and this project is complementing the important EUNet CCC project. So from the point of view of from the Cancer Mission, I really think that we are trying to overcome these barriers which can exist, especially between the member states.
Mm-hmm, mm-hmm. So we've started the discussion on how to translate the laboratory discoveries into clinical use, of course, and we were talking about money, we were talking about scaling, and the problem with patents, really, the will to invest into something where you can't patentize it. But Mr. Aguiar, you actually are translating into clinical use, aren't you? You have experience with that.
Yes, and I think it's the same what for the previous speakers mentioned. It's too high. Sorry, it's too hard. Of course, to run a high quality research, it's not maybe so problematic, because now we can have especially across the Europe, the very high quality research infrastructures, which is also the case of our country. But the problem is of sustainability of such research, because this research is producing new questions and new ideas to work on it. And if you discover something, then it's the problem to transfer it to the clinical practice, because it's one of the most important factors to how to develop such drugs or molecules on the level which is able to use for clinical trials, even if just for the first in human trials. So that's a problem, and we should also work on it and think on it how to address this, because yeah, the problem of the commercial pharma companies, which are helping, of course, and are sometimes give us some opportunity for investigator-initiated trial. This is not the right and complete way how to transfer the research to the practice.
Right.
Sometimes it's not so problematic, especially with using the biomarkers. On the other hand, all these indications in the clinical practice for the immunotherapy is done on evidence-based, fortunately done on evidence-based. And also for such biomarkers, we need to do very comprehensive biomarker clinical studies. It's also not very easy to run such trials. So support for this could be also very important, and support which will be crossing all the member states. Yeah, there are some difficulties, of course.
All right. Thank you. I was actually going to ask you, Dr. Aguiar, about the translation of the lab, of the whole lab discoveries and into clinical use, because to be honest, right now it seems, I mean, to me as a layperson, I'm thinking, well, isn't it just early days? Isn't that just the problem? Or what, when it comes to immunotherapy and the precision treatments? What's your experience?
No, what we see treating patients with TIL therapy, when patients respond exceptionally well, is usually because they combine some biological conditions at the same time. These patients often...
Which ones, for example?
These patients, for example, often have tumor microenvironments that are already permissive.
Yeah.
For example, they have high levels of pre-existing TIL infiltration, and importantly, those cells are not completely exhausted. So even if we have tumors that are very aggressive, we can build an immune response using this type of therapies. On the other hand, in non-responders patients, we usually observed profound immunosuppressive microenvironment with either no meaningful TIL infiltration, or with TILs that are so dysfunctional that expanding them doesn't translate into clinical activity. The second point is that I think biology is important because responders tend to have tumors with high new antigen burden, high clonal antigens, which makes them more visible for the immune system. So when we expand cells for these patients, we are giving true cells with a true tumor reactive potential. So that it's important. In non-responders, we usually don't observe strong new antigens. The disease evolves very quickly and escape the immune system. Another thing that it's interesting is the architecture of the tumor. We are learning that the special organization of the TILs, I mean, the capacity of the TILs to physically achieve the tumor cells is as important as TIL cells themselves. Responders, as we mentioned before, usually have more inflamed tumors, more accessible tumors with less fibrosis, like the pancreatic cancer, for example, and less signs of immune exclusion, for example. And finally, I think that the patient-related factors are also important because some patients recover faster from the lymphodepletion, are capable to tolerate more doses of high-dose interleukin-2, and they are able to maintain these expanded cells longer and make this type of therapies more effective. So I think that the main important thing is to select better these patients, and that's the reason precision immunotherapy is very important in this case.
Let's talk a little bit more about the approach, but I want to tell you we're going to have a Q&A session again, and I think especially this panel there could be a lot of questions, so I invite you to start asking your questions now. It can be done on Slido, just like in the first panel session that we had, and please just start them coming and keep them coming, and we'll get to that shortly. Let's return a little bit to what you're working on, the intratumoral administration. this approach is, of course, completely different than the systemic one. And you explained how it is. I mean, you go directly into the tumor instead of the systemic. But why is it that you think, or you have proof that, I don't know how you word it, that this is a better solution altogether?
Well, the funny thing is that historically the chemotherapy started from the intratumoral administration. I don't know if you know that.
No?
Yeah, the first chemical approach to treat the tumors was done in the 40s, in 42 actually, in US.
And they would inject chemo into the tumor?
Yes.
Oh.
There was a patient with lymphoma, and he was treated with the mustard derivative, mustina, by the US military.
And why did they stop doing that and start on the systemic approach instead?
Why? Because this approach was only limited to the local activity. There was no derivatives, no immunotherapy as we are having right now. So for the next 80 years, all the approaches were done through the systemic administration, because the idea was, okay, with the blood, we can deliver something systemically. We can act on the metastasis, we can act on different infiltrated organs, not just locally. But this led us just to thinking about the systemic approach as the only one approach. Right now, with the immuno-oncology and immunotherapy, we are coming to the point that intratumoral administration might be much more feasible. And there are several reasons for that. With the systemic approach, we were creating the problems ourselves immediately, because there was discussion, okay, so what is the biodistribution? Will my product will go to the tumor or not? What will be the systemic toxicity? Because if I'm delivering something, I have made a bigger titration, bigger number to get something at the tumor while the other organs were affected. With intratumoral administration, we do not have this. Because first of all, we are giving something locally. Second, we are using a very small dosage in comparison with what we are using in the systemic approach. And this is starting to turn out to be visible in the approach. There is already one big pharma, Johnson & Johnson, has already created a separate team just for the intratumoral administration. Why? Because of this development of this kind of the approaches. You do not want to create billions of the lymphocytes to go into systematically if you can deliver them locally. It's the same with our approach. We do not need to put billions of the macrophages in systemically. Even we know the macrophages are going to micrometastasis, and it was a shock for us a bit when we have delivered to the animal and they were located in the micrometastases which were not detected normally in the PET. And histopathology, each one that they are going there. But anyway, intratumoral means that we are activating immune system. It's not the drug, it's the immune system of the patient.
But it is too when you have this systemic approach with the immunotherapy, right?
Yeah, but with systemic approach, we are delivering product, and most of the therapies actually are molecular target dependent. So this is why we are talking okay, there is an antigen, okay, the tumor is elapsing, the antigen is disappearing, it's heterogeneous, and there's always problem,
Right.
Why checkpoint inhibitors are not acting in 100%? Right now, it's up to 30% of the patients are really responding to this best immunotherapy product. Why? Because of the changes of the tumor microenvironment, the antigen escape, heterogeneity, just tumor microenvironment, and
Is this controversial?
the intratumoral approach? Absolutely. When I'm meeting, and I'm really meeting lots of VCs and lots of big pharma, it's always, well, no, we are not in this. No, we are not in that intratumoral. We are in the systemic. No, we are not in the cell-based therapy. The most annoying, I would say, is the approaches of the VCs who are saying, it's interesting. But we would like to have a product which is second or the third in the class. This is what I heard from the European VCs. It's amazing because these people will never back revolutionary approaches. It's sometimes you have to be bold. You have to be brave in your thinking.
We're not going to talk badly about them because they're not here to defend themselves, but I got your drift and I asked the question, so thank you for answering. Okay, I think actually a few questions are starting to pour in. We're going to wait with them just a little bit because I do want to talk a little bit more about what it would require to get further ahead, to speed this process up, developing precision immunotherapy, since whether it's in your type of approach or the systemic approach, either or, we need to speed up the process, and it requires money, and it requires scaling, it requires also a lot of collaboration, not least between sectors. And it seems like that's not always the case, that it's able to do that. Maybe someone is conservative, we don't know about that, but maybe. Maybe there are other obstacles in the collaboration needed. So I want to ask you, before we dive into the questions from the audience, how do you, just based on your everyday view on how the different sectors work together, collaborate, how do we build more integration across the different sectors? And we'll include you here, Professor Svoboda. You sit at an intersection of national governance and research. And from that vantage point, what is the biggest barrier to the necessary collaboration across sectors? And what do you see as the biggest barriers to that, for a start?
Thank you for such a question. It's difficult, but before I will answer, let me add something. Let me comment on what was said as the last, because I think that it was very, very important. It shows us that it's very important to build or to have a good infrastructures, research infrastructures, which are capable of production of these new ATMP drugs. Because, for example, I remember in 10 years ago, or maybe 15 years ago, we were firstly applied dendritic cell vaccines into intranodal in the patients of the melanoma, and it was actually in some cases effective. So if there is no willing of the pharma companies, so these big players, to work on such approach, which was mentioned already, we should really have we should really provide a support how to keep such capabilities in our own hands. For example, like it's done in Fraunhofer Institute in Brno, we have also a creatic center which is able to produce such vaccines and apply locally. But going back to your question, that's as I said, it's difficult as the situation can vary significantly between the member states, as healthcare and the research are there in their exclusive policies, and it is therefore very important that there is a unified policy for the fighting against cancer in the form of the European Beating Cancer Plan and the Cancer Mission, as well as a good coordination between the research and the healthcare at the EU and Commission level. An example of this, it could be, for example, the Cancer Mission Board, where various stakeholders are representing, and which serve both DG RTD and DG Sante as an advisory committee. And this is a very good basis for inspiration at the national level, if I understood well the question. This is because we also need to communicate between the various stakeholders at the national level in order to at least highlight the importance of the research and also to create synergies for financing research projects that can support the projects funded by the EU funds, but especially, which is very important from my point of view, to maintain high quality research infrastructure at the national level, especially those which are a part of these pan-EU infrastructure network, like for example for biobanking, translation research, or for example for conducting academic clinical trials. So personally, for example, I have a very good experience in my country and also in the neighboring countries, like for example in Poland and Slovakia, with the national contact points for the Horizon Program Europe and the Cancer Mission. And also I firmly believe that the potential of cancer mission hubs will also be used for this purpose. Because that's very important to synchronize and make the synergies on the national level, because there is a source of financing which we can use for this purpose.
All right, Professor Birg, what's your experience with cross-sector collaboration?
Well, like my experience. So I work in the university hospital as well, and we used samples from the hospitals. That's what we build a lot of our research on. But the issue I often run into is that clinicians do not think research, and that's something that I think needs to change. But it also means that the management of
They don't what need?
think
in research, right?
They treat patients, which is their primary job, but they also need to think research into their everyday work. But that the management also needs to support this because it takes time. And often they are just overworked.
And in a hospital ward, they think productivity, and they have a lot of other things on their mind.
They need to treat the patients, which is their primary concern. Of course, it should be.
Yeah.
But they also need to think research, because research that improves the treatment in the end. Just the fact that you have a research program means that you optimize different processes because we need different things.
So this is a bit surprising to me, but although not complete. Has this changed? I mean, has it changed with increasing demands on productivity at a hospital and so on and so forth? That we've moved away, the clinicians have moved away from thinking in the lines of research and
there has been, at least here in Denmark, and probably the same throughout most of Europe, there has been increasing demands on the healthcare system, and without funding to match.
Yeah.
And they are generally extremely busy and overworked. There is now a push to make most of our hospitals university hospitals, which means that they should think in research. And it does improve within the past couple of years. I have experienced that. The infrastructure, as you mentioned, Dr. Merck, has been generally, there's a push to build this infrastructure and to improve the access to research, so to facilitate collaboration between universities and university hospitals, to directly allow access to patients. But of course, here again, we run into these regulatory hurdles that take some time to get across, because we need data sharing agreement, etc., even if we are in the same setting. But it is improving, but I think this is, from my perspective at least, one of the main issues is that clinicians simply do not have time to think in research.
Right. I want to ask you, Dr. Aguiar, because you're actually leading a mission project that does bridge academic research and clinical implementation, as we've talked about before in this session. Are you a best case study on that? You can say yes.
Yes. So we needed to think about that in the day one of the BRAGMA2 trial, because as we mentioned, you mentioned before, researchers think in terms of academic questions, clinicians think in terms of patient experience and urgent results, and regulatory bodies think in terms of safety and so on. So in BRAGMA2 trial, we incorporated different two products. It's very interesting because each product was manufactured locally, but each participating country, and was approved by each regulatory agency. So it's true that we observed some slight differences in the manufacturing process, especially in the expansion process of the TILs. But the data shows that these differences don't impact the safety or activity of these products. We know that in TIL ACT therapies, The toxicities come more from the lymphodepleting chemotherapy and the high-dose interleukin-2, not from the TIL product itself. So during the regulatory revision, the agencies asked us to justify why we are using different TIL products. So we put the non-clinical, clinical stakeholders together to address this question. So we prepare documentation of the non-clinical, preclinical results and comparable clinical results. And we also prepare a comprehensive manufacturing process document to address that. The regulators ultimately approved the protocol and the clinical protocol in the project, and they recommended to continue collecting this comparative data. So we included this recommendation in our statistical analysis plan as well. So in conclusion, I think that PragmaTIL trial embraces this pragmatic, resource-efficient approach because maintaining the institution-specific products avoids us all the standardization efforts that could impact cost and time. And with this approach, I think that we can accelerate the patient access of this type of therapies in Europe.
So you can say you kind of massaged the science or the research into the process from the beginning.
Yeah, and the European Commission, the European agency agreed with that, and we have this project ongoing. So I think maybe it's a way to expand the access for the European patients of this type of therapies, especially in the academic and research centers, because we can't escalate like the industry. But if we follow the same manufacturing processes, we could include more patients and be able to give them this type of very personalized treatment.
All right. Let's move on to taking some of the questions that have floated in. I think we can start with the latter one. Are there known examples of natural spontaneous regression linked to rare functional immunological responses that can be expanded on? Are there known examples of natural spontaneous regressions linked to rare functional immunological responses that can be expanded on? Okay, who wants, this is like in school, who raises their hands to answer that?
You work with biomarkers, so.
I think the answer is probably, yeah, there are, I'm sure. I don't have specific examples, but the difficulty is to link them to functional response that we can actually use.
What is this person asking about?
Because
for everybody here who's not
asking
researchers.
if a person where the cancer goes away spontaneously.
Yeah.
If we can learn something from that. And the answer is yes, we can. But it's very difficult to expand that to the general population and see, okay, the tumor disappeared, what is there, can we expand this to others? I'm sure this has happened, I don't have specific examples, but tumors do spontaneously regress occasionally. And often the explanation is something like there was an immune reaction against something. We do not know what this is. You usually need a larger cohort in order to really establish it. We can build a hypothesis, but it's very difficult to generalize from a case of one. I can.
Are there ever, some of the, you can say, crazy hypotheses built on observations like that? A lot of people think that if they eat, I don't know, carrots or whatever, that will help their cancer go away. Are there ever any of the little, crazy hypotheses built on stuff like that?
Area of expertise, but it's sure it happens. and often it can be linked to some kind of infection. So I think that was shown more than a century ago by a famous German, I don't remember his name, who ended up injecting live bacteria into tumors, and they also
Yeah.
regressed.
Like we're also going to hear about later today.
Yeah, so it's often some kind of mobilization of the immune system for reasons that we do not entirely know.
So something coincidental, and you latch onto it and
Yes, we
try to figure out.
there are lots of aspects around the immune system that we really do not know anything about.
Yeah.
And this is one of those things that happens. And we are trying to use that with immunotherapies. This is what immunotherapies are based on.
Exactly.
Mobilizing an immune response through the cancer itself. And we have a few angles on how to do it. This is the checkpoint inhibitors, the CAR T cells. There are undoubtedly macrophages. There are undoubtedly many other things that we can do, many other aspects that we do not know about. And that's also because the immune system is incredibly complicated, and I think we have not even scratched the surface on what the immune system is capable of and how it interacts. There are at least 10. Yeah, go ahead.
You said a very good thing about it, that immune system is very complicated. And sometimes we should be, as a human, humble, I would say, because we try to figure out to the very extent what is the mechanism. And sometimes we should come like IT people are coming with the information system. Black box. Sometimes I'm doing something, as you said, there is some activity.
I know that from my IT support. He says, it's working, don't fix it.
Yeah, exactly. You're doing something and you have the effect. And if it's repeatedly happening, then, okay, you can spend time figuring out what's inside the black box. Maybe there are dwarfs who are doing something or whatever. Yeah.
But is that research?
It is a research. It is a research. From the clinical point of view, for the doctor, it is important that the patient is cured. We might not know to the 100% how it's happening, but we know that if we give one particle, we will get this kind of the effect.
But don't you need to know why it helps in order to recreate it?
We should be humble. Yes, of course, we should be curious. We should be scientifically curious, to investigate. But it should not stop us to use this kind of the process. It's working. Okay, 1 second, 1,000 times, perfect.
What do you think about what Dr. Alexandrovich is saying?
I completely
here?
agree. And also that it is research to try and figure out. That's what we mostly do actually. That's trying to figure out the mechanism. Why does this work? This black box, taking it apart. But there are so many ways to perturb the system that it's. So this is going back to my scaling thing, that we need a lot of data in order to be able to really discern how does all of these different cells interact and what is the impact of age and sex where the immune system changes. So your immune system is different from how it was 20 years ago. And the effect of your pill will be different. We don't know how. We don't know what the changes are. My lifestyle will also impact this. But we can tell that it works on some patients. Can we make a biomarker that will identify these patients? Maybe that's the angle.
What's your thoughts on this approach?
I usually don't see spontaneous responses for the patients, but it's very interesting when we have in a clinic a patient with a long-term response, even without treatments. In Valdebron, we have a molecular pre-screening program, so we try to understand, collect samples, try to understand what's happening with these patients. Sometimes we get some information, sometimes not. And even if we find some molecular, biological characteristics, each patient is a different patient. So we can't translate the same situation for the other patient. Even if they have the same tumor in the same stage, you give the same drug with the same schedule, it's very difficult. For example, the CAR T cells in solid tumors, it's very challenging to produce CAR T cells for solid tumors because sometimes the tumors have different types of new antigens. So you target one, but you select this population, and in the same tumor you have other population with a slowly growing. So in a couple of months, years, the tumor come back. So it's very hard. Not the immune system is very complex, but the tumors are very complex as well.
Okay.
Can
Yeah.
I add
Yeah.
one
Yeah, you can.
point? Because again, I would like to everybody to be aware of one thing, how the big pharma is thinking and what is really changing the healthcare system. We have been talking here at the very beginning. 3 million patients in cancer patients in Europe. How many patients we are using in the clinical early stages? 50? 100? Imagine that your technology is working. Bravo, success. We have tested it on, let's say, 500 patients. And you have 100 clinics across the Europe which is producing this. And we have 3 million patients, maybe 1 million, let's say, with this indication. And this is the challenge. You know, the big pharma is coming and saying, okay, how do you want to scale up this? Yeah. If it's a CAR T, how do you can afford to pay 100,000 or 500,000 by one administration or per one patient? It's not feasible. So the thinking about the translation of the great research should always include this element of money.
Back
Yeah.
to money.
And,
Sorry, but I have to be
No,
straightforward.
and another thing that's very important in this case, for example, that's the reason that it's very important to support the research in academic
Yeah.
hospitals as well, because
Yeah.
sometimes in these cases, when the industry, for multiple reasons, are not interested to treat patients because they have a rare disease, because it's difficult to find these patients, maybe there is a room for the academic centers to treat these patients. So regulatory pathway would be important to put the industry and academic centers together to reach these patients and reach
Well,
these
maybe you
unmet-
could say
unmet needs.
ultimately we end up in an entirely different discussion talking about ethics and money and a lot of other things and when should you end your research and so on and so forth. We will not go there right now. We'll return to the questions and it's elaborating on what we've already talked about because somebody's asking if we can explain how a research program optimizes the clinic. Implementation takes a long time. What key steps would you take to connect research and the clinic, which is essentially one of the large questions this entire day, to be honest. And we've touched on it before. We need the willingness to invest. We need to scale, to be able to scale. Maybe we also need a different sort of collaboration between sectors because if those are detached from each other, nothing's really going to happen. You can't just view the clinicians as end users and nothing else. What else?
The best, I think, sentence, the fragmentation of the efforts. Developing from the research to the final product is the process. Yeah. You have mentioned about, this connection between the hospitals and the research. The same connection is necessary between the research and the industry because everybody is going to, and the same is question of the startups and the VCs and the financing.
But how do we connect them better? Because we can all see that this is too fragmented. It's not doing any good when it's so fragmented. Well, maybe some good, but nothing that I can view in terms of speeding up the process of bringing more into the clinics and faster. But how can we speed up that process? What can we do to unite all these factors?
I'm wearing, sorry for this,
Money.
I'm wearing for the businessman hat on this discussion. But it's really about the quick support of this kind of the research with the risk funding. Yeah. And we are really not having enough the risk funding in Europe. Mm-hmm. There's plenty of money inside the pension funds, insurance companies, the banks, and so on. Mm-hmm. You know what kind of the questions are raising right now? The VCs, which are supposed to be a risk money for this kind of the program, they are asking you, okay, show me the phase 2 data. So after the clinic, when you will be ready for the IPO, will you be ready for the M&A transaction in 1 or 2 years? Of course not.
No.
Because
But that's the
I'm
nature of the
here,
VC.
you know, I'm
That's the
struggling.
nature of VC.
Yes, but we are lacking this. And
They're
there's-
cowboys.
They are not the Cowboys, maybe Cowboys, not the Cowboys, but the Europe has to shake up the financial institutions. Without the flow, the money
back
to
up
this,
slow money, yeah.
we will be just talking about the great research, but it will not help our patients, never, because it will not finish in the industrial level.
Anything to add?
Well, I'd like to answer that question,
Yes.
actually, because I think this is an important question. So when a research program is included in the clinic, so this involves collaboration between academics and clinicians, and this makes the clinicians think research. So they need to read the papers, they need to understand what's the research project about, and then a group of patients will be following a research program, which means some kind of standardized pipelines, state-of-the-art technology is being brought in. The whole department will start to recruit patients, and then suddenly patients that are not in the program will also be receiving this, not necessarily exactly the same protocols, but they will be receiving the same kind of surveillance, same kind of care, and this makes the clinic improve its treatment. This step alone makes the clinic improve.
We don't have any clinicians here on the panel to reflect on what you're saying. I'm just saying because this is that could have been interesting, but that's the way it is right now. Yeah.
Yes, I agree. No, I agree that I think that we need clinicians that think as researchers as well.
Yeah, but you are both, right?
Yeah. Because what we see, for example, in Waldebron with the TIL program, we have other clinical trials in TIL program, not only the PRAGMA-2 trial, but we notice that the clinical trial changed the culture of the clinicians in a way. For example, now we have the ICU physicians dedicated to understand the consequences of TIL therapy, the adverse events when this patient, for example, when we give high dose of interleukin-2, sometimes we need to transfer these patients to the ICU because they need support. Before, these physicians thought, okay, this is a patient with a metastatic disease that is progressing and is receiving an experimental drug. So maybe we should only give a support and let the nature do this, this whole. So now they understand that sometimes we send these patients there because they have some adverse events related to the treatment and they can recover and they can have responses from this treatment. So we now, for example, have cardiologists, rheumatologists dedicated to treat patients with immune-related adverse events to try to understand that. So I think that research brings in the clinic some changes in culture, but I agree that clinicians need to start to think as researchers.
And when I say we don't have, it's just because I'm thinking not in the line of the one person who's in the clinic. I'm thinking in the line of the systems that we have built up on around ourselves, the hospitals and how they're working and what task is it that they're trying to solve there, as opposed to what you're doing in the lab. Just to clear that up. Okay, we're approaching a wrap up now, and is there anything that you'd like to add to what we've talked about? If the broad task here is to talk about how to speed up the process of understanding how we get further with precision immune therapy.
I will think that about this last question, how is EU Cancer Mission specifically supporting progress in immunology and biomarker development? I think the Cancer Mission is positioned very well in the way that it should, again, about the cooperation we are saying, cooperate with the other EU programs. There is a program like InvestEU, which is absolutely financial program. There are other programs which could be linked. There are these discussions after Mario Draghi report, changing the capital market. You know what was the problem with the VCs in Europe? They are complaining that there is no single capital market like US Nasdaq, where they can put their companies and they can return their money.
They need scale as well.
Yes, but these are this. If we are talking about the cooperation, you see, we can talk about the cooperation at different levels.
Yes.
Really, a huge levels at the European Union, and that's necessary to scale up things.
I wouldn't think it.
And regulatory approval.
Right.
So you need to adjust this massive layer of bureaucracy and ease the regulation that we need to follow to make a path that we can collaborate across
Integration,
borders.
yeah.
Yes.
Okay. Professor Svoboda, do you have anything to add to what we're
Yeah, I
talking
have
about?
two comments. First is to the interaction of how we can straighten the interactions of the clinicians and the research. I'm pretty sure that we must make bigger room for academic research, but especially, and this is our good experience, with dedicating time to the research. It's of course not very easy done, because the number of patients is rising. They need a more time dedicated for the consultations. There are much more multidisciplinary team discussions, and also the secondary opinions. But on the other hand, we will adapt this research to the career paths of these clinicians, and if it will be some kind of duties for the career growth of our colleagues, it can also a little bit fasten this process and make it more effective. The second, what I want to comment, I just find some information here which are really very fresh. The Cancer Mission really support the research on the biomarkers. It was one of the questions. I just find that the last call, which was closed, let me check the data. It was closed just in September. There were 116 proposals, and one of the Horizon Mission Cancer topic was pragmatic clinical trials to optimize immunotherapeutic interventions for patients with refractory cancer. So I believe it will also help to solve the problems we spoke today about. And finally, I would like to give you some kind of idea, or give you the example of a very good website when you can find all the informations of the cancer project supported by the Horizon Europe and Mission on Cancer Calls. It's the website called Cancer Projects Tool. It's absolutely perfect website with very good infographics. You can find all the calls which are running, all the projects, and their interaction between them. Because sometimes there are many calls and we have no idea, do they collaborate or not? Do they share the results? Please go to this website, the Cancer Projects Tool.
Cancer Project Tool. All right.
Yes, Cancer Project Tool. I will put it to the chat box.
All right. This has been brought to the audience here. Professor Svoboda, thank you so much for participating from afar, and thank you for participating. Thank you for delivering all your enthusiasm and knowledge, and just for being here. Thank you very much.
Thank you.
So this was an interesting and long and thorough panel, and I think now we need some lunch. And the lunch, fortunately, is ready in 30 seconds. So I think we have just time to walk out there. And the lunch is served in the restaurant, and not in the back where the coffee is at. And we will convene again at 2 o'clock. So there's a good time, lengthy time to network and have a bite. Enjoy.
Introducing the all-new EU Funding and Me app. The app that transforms the way you search, manage, and make the most of your EU-funded projects. Combining the thousands of opportunities on the Funding and Tenders portal with the ease of use of a mobile application. Save your searches, bookmark your favorites, and never miss a deadline, event, or news item thanks to personalized notifications. Boost your knowledge through funding-related news and info days with in-depth information and insights. Coordinating EU projects? No worries. The app makes it easier to reach out and stay connected. So, if you have an idea, Eureka, download EU Funding and Me.